in my library. So I finished that. And then I had one this last class.
Or those papers that go to do it? Hey everyone, thank you for doing that rearrangement. So I think, if I'm not completely mistaken, this is the second from last class, right? I think the last class is on Tuesday. Okay, so, and I know that, end of semester gets very busy for everyone. Anyway, so we're going to talk about a bit more examples of applying fMRI and pattern recognition to real-world problems. I'm going to finish talking about the dyslexia study that we started looking at a little bit on Tuesday. We're going to talk about some interesting studies detecting consciousness. First of all, just a few quick project notes. So the first thing I want to say, actually, which is not on the slide, but I should say, is don't panic. Don't worry about, my aim of this project is not to make people get stressed or upset or worried or anything like that. And, I should probably have some kind of explicit policy in place about, exactly what happens if you hand it in, like, late or something like that. So I once took a class and the person had a policy that for every day it was late, the grade of the thing decayed by 10%, which I thought was actually kind of a good policy. But, you can't really announce that just like 3 days before the thing. So, I'm going to say, please do hand it in on time, but, if it's a choice between extreme panic and mental distress and handing it in a couple of days late, I need to be able to submit the grades in time. And so if you hand it in, many days late, I might not be able to do that. But I don't want anyone to get like stressed or panicked or worried. So please hand it on time. The sooner you hand it in, the longer I'll have to look at it, and so the more attention I'll give to it. But don't panic. Don't stay up all night doing it, or stuff like that. Yeah.
Do you want us to just email you?
Email is good. Yeah, email is good. If I think I mentioned like a previous one, if it's a really, really big thing, which it probably won't be, probably text is fine. If it's some mega huge file, then maybe send it as like a Dropbox link or something rather than attachment. Yeah, email is good. To like my Gmail, the one I've been sending everything from.
You think?
Yeah. So please, please do your best. Please hand it in as on time as you can, but please don't have a great degree of stress or mental anguish about it, because that's really not the point. And by the way, I didn't mention at all any of this, but there's lots of interesting studies being done of sleep and the brain, and it turns out that if you don't sleep properly, your brain is completely unable to consolidate new memories. So this is, by the way, one reason why you shouldn't stay up all night in front of before an exam. So I don't want to like contribute to people having to like miss on sleep. But so the closer to Tuesday you hand it in, the more I will look at it properly. And if it comes in much later than that, I might not get to look at it properly. But that's so that's kind of maybe sort of quasi-equivalent to this thing about decaying grade. Another thing, and a couple of you have given me like early drafts, and you can give me. I mean, you can give me if you want, just like here's my opening paragraph. What do you think? Or something like that. One of you has even given me a second draft, which I haven't given comments to yet, but I gave, which I will do. Sorry. So it's not like a one-shot process too. You're welcome to run any kind of little snippet by me if you want. Although I will actually be traveling this weekend, so I probably won't be emailing much this weekend. One thing that has come across from just looking at a couple of mini drafts or just chatting to people, and I sort of mentioned this before, but I'm just going to mention it again because it's come up again, is do not assume that the reader cares about your favorite brain area, or even that they care about the brain at all. So imagine you're talking to someone who cares about human beings and cares about, knowledge and science and stuff like that, but didn't wake up in the morning thinking, man, I really want to read about fMRI scans this morning. Because this is actually one of the problems with the field of imaging in general, is that, say a sort of like what lit up paper might be of great interest to somebody who woke up that morning wanting to know, will this task activate, the supramarginal gyrus or something. But for most people, there's no particular reason why they would care about that. Now, it might be that after you explain, well, actually, here's why I actually care about this type of task, or this type of condition, or this type of, circumstance in life that affects people, or that affects animals, or that affects any cognitive organism. And it turns out that this region is incredibly strongly involved in that. And if we really could understand this particular question and how it relates to behavior, then that's why we'd want to know about that region. Then that's fine. Okay? But if the opening line of your project is something like, this project is gonna, explore the properties of the supramarginal gyrus, then that will immediately lose anyone who didn't wake up that morning thinking, man, I really want to learn more about the supramarginal gyrus this morning. And similarly, on a similar note, try to avoid vague phrases. Try to, be as specific as you can. I know I'm not telling you anything you don't already know, but this is just sort of come out in various projects. So try to avoid things, especially in your opening lines, things like explore the role of, or the neural basis of. And a particularly bad one, actually, in my view, is we predict an interaction between. Often, suppose you wanted to say, I'm going to Boston, right? You wouldn't say, I'm going to the East Coast of America, right? You would say, I'm going to Boston. But there's a sort of disease that affects all of it. It affects me, it affects everybody else. There's sort of a disease, especially when you try and write stuff down, that makes you somehow think it, not you, this is just true of human beings. We all have to fight against this. Somehow there's this great pull, it feels more formal or more scholarly, I don't know what it feels like, to say, I'm going to the East Coast of the United States. No, if you're going to Boston, say you're going to Boston. Okay? And so, I was looking at a draft project that was actually a wonderful project. It had great ideas, it had great specific questions, but it started off with a very vague sentence about this is going to explore the role of some area. And by the end of page 2, I found that there was actually really interesting stuff going on. But so try to avoid that. The reason why we predict an interaction between is terrible, because usually what that means is I think that X is going to go up for this reason and Y is going to go down for this reason, right? So if you think X is going to go up, say X is going to go up. Don't just say we predict an interaction between X, Y, and condition Z or something, because 100 different things could all meet that criterion. That's like saying, I'm going to travel to, the northern hemisphere instead of saying I'm going to Boston. Okay, so those are just some comments on that. If you want to meet with me, then I'm scheduled, I think, to be meeting with at least one person after class today. I have time, I think, to meet with 2 people, so if you'd like to meet with me just after class now would be a time that could work, or we could also do it on Monday, potentially. Okay, let's talk a bit more about some specific applications that I personally happen to be interested in, but I think, my hope is that other people find them interesting too. And I think what's particularly interesting about them is that they go beyond what you can get from behavior. I know I go on about this a lot, but I really think that, what is the point of sticking someone in a scanner if you're not going to get something over and above what you would have gotten if you didn't stick them in a scanner? And that sounds like an incredibly trivial statement, but given how much you can get without sticking people in a scanner, it actually becomes a non-trivial statement. It's amazing how much you can get from behavior. And many mechanisms, many actual neural mechanisms were found using behavioral measures, which is kind of amazing. So people found things like the existence of spatial frequency channels in vision just by, showing people it wasn't even a computer screen, a luminescent phosphorescent screen, and getting them to push buttons. They didn't have to stick any electrodes in anyone's head or put anyone in a scanner. So this is a nice paper that shows something that goes beyond behavior, and I'm going to show another one in a minute too. we started talking about this. So this is not a very good slide because it's got too much text in it, but I just want to, this is literally just quotes from the abstract, but I think it does highlight some of the key points. So first of all, it's an actual real predictive study following people over 2.5 years. It's the easiest thing in the world to fit something to some data and say you predicted the data. It's much harder to actually literally follow people over a long period of time and actually predict outcomes from what you measure at the beginning. It doesn't happen very often. These types of studies are difficult, expensive, require lots of practical difficulties tracking people, but they're very valuable when they're done. And so the aim is to predict long-term reading gains. Look at their brain at the beginning of this time and say, hey, I think that this is going to happen or that's going to happen, and see if you're right. And what's especially interesting is that they also had a whole bunch of behavioral measures, and the behavioral measures were interesting and valuable, but did not predict future reading gains. So you measure someone's behavior, and you can say, okay, they have reading problems. How are they going to be in 2.5 years? I don't know. And so it's interesting in terms of this class because they did some pattern analysis, which I'm going to show a bit more in a minute, and that actually turned out to be quite good at predicting. And so the punchline is, so this is the why should you care bit, right? Okay, the why. I know I keep on going on about the why should you care, but if you never explain anything to anybody except why they should care, you'll probably be in quite good shape. That matters much more than anything else. So why should you care? Because predicting future behavioral outcomes, neuroprognosis, may be more accurate in some cases than available behavioral measures. There's a famous phrase, I'm sure you've heard it, that is sometimes attributed to the physicist Niels Bohr and sometimes attributed to the baseball player Yogi Berra. It might be both of them. Prediction is difficult, especially about the future. Okay? Everybody wants to be able to predict the future. Predicting the future is very, very difficult. So if you can find anything that gives you a little bit of extra information, that can be valuable, especially if it's something that affects millions and millions of kids. So here's their subjects and what happened to them over time. So the first time point is the beginning of the study, second time point is 2.5 years later, and they're measuring, what are they measuring here? Single word reading, and some comprehension score. Okay? And so the black line, which is way up at the top, is normal readers. So they're doing great at the beginning, they're doing great at the end. That's what you'd expect. And then they have people who are much lower down. Notice that 100 is sort of the average. So typically to count as dyslexia, you have to be a standard deviation or 2 below the average. So that's where they all start off. So everyone starts off really low, and some of the people do better over time. These are these red lines that creep upwards, and some people don't really improve. These are the blue lines that are kind of flat. Okay? Now obviously that split into the sort of improvers and non-improvers is a little bit arbitrary in the sense, it's not arbitrary because you're either up or you're down, but it's taking something that's continuous and sort of dividing it into 2 chunks. But dividing people into those who got better and those who didn't is not a terrible way of carving up the world. And so they asked the question, can you, from your behavioral measures or from your neural measures, predict at the beginning, using this data from the beginning here, whether one of these poor readers is going to be in the red line of people who actually get better or the blue line of people who don't? And so they used a classifier to do that. And we've talked a lot about classifiers. But it struck me, and I look back, actually, I don't think I actually went into any sort of discussion of a particular type of classifier, which is the one that they use, which is actually worth knowing about. I mean, many of you probably know this already, but I'll just say it anyway. So we talked about all kinds of classifiers, and basically what a classifier tries to do, or at least a discriminative classifier, is it says, okay, I've got 2 groups of things, the red dots and the blue dots in this case, and I'm going to try and draw some line between them that says everything on one side of the line I'm going to call blue, everything on the other side of the line I'm going to call red. And the problem is that there may be lots of different ways of drawing that line. And you may not even want to draw a straight line, but even if you draw a straight line, so you're really kind of tying your hands that way, you're doing a linear classifier, there's still a bunch of different ways you can draw it. And that's nice. I was just Googling for like pictures of, I mean, you actually can't see this right now, but you'll be able to get it from the when I upload the PowerPoint. But I was just Googling for nice pictures illustrating this, and this website actually seemed to be quite good. I have no idea who this person is, but they made some nice figures, and they actually explain things quite well. So, you know, now it's very intuitive and natural to say, oh yeah, you know, this green line sort of looks better than this red line. Okay? Well, what does sort of looks better mean? You know, a lot of machine learning could be crudely paraphrased as saying, try and get a dumb, mindless computer to figure out how to carve up the world in a way that sort of looks right and makes sense to us as human beings. And, well, certainly this green line sort of looks better than the red line. Well, why is that? Well, one way of quantifying it, and it turns out to be a very good way of quantifying it, is to say, suppose you take these candidate decision boundaries and you say, how far away are they from the nearest points? In other words, how big a margin do you have between the boundary and the kind of nearby stuff on either side? Now, some of you are probably thinking, well, hang on a minute, you're not going to have any margin at all if these are actually overlapping classes, and there's ways of dealing with that, but I'm not going to get into that now. So what this picture nicely shows on the right-hand side is if you actually draw in those little boxes of, you know, the biggest margin you can get if you go away from the boundary to the nearest points, then sort of fitting with your intuition, this green line, which sort of looks like a better line in some intuitive way, in a very quantitative way, actually gives you a bigger margin. Okay? And so not only is that better because it kind of looks right, but it means that you literally have more wiggle room. So when extra stuff comes in, you're actually more likely that the new stuff is going to fall on the correct side of the line. So you will actually generalize better. I'm expressing this in very hand-wavy terms, but there's a lot, if you're into this, there's lots of mathematics behind it. A guy called Vapnik figured this all out. So you will actually generalize better because if you've got a big margin, the new stuff that comes along is more likely to fall on the right side of the line. So these boxes show the size of the margin. So that's what an SVM is. SVM stands for Support Vector Machine, which is a very weird and mysterious sounding title. It turns out that these points here that are the ones that are right on the margin, those get called the support vectors because they kind of support the margin because it touches them. But it's not a very intuitive name for it. But anyway, so that's what an SVM is. The people use them a lot because they work very well. For a long time, they were sort of like the best working thing in machine learning, and now that's sort of been overtaken by a much more complicated approach called deep learning. But they work very well, and they're not that complicated to get running. So you've got your people who improve, you've got your dyslexics who 2.5 years later get better, and you've got your dyslexics who 2.5 years later don't. And if you just sort of like slice up the world into these two groups, and you say, from my data, my fMRI data, my behavioral data, can I classify people as belonging into one of these groups? So this might be the people who get better, and this might be the people who don't get better, and you're going to try and carve up the world in the space of neural data and behavioral data, and you're going to try and do that with as big a margin as you can. And then what they did was, what's actually a bit confusing is that these graphs look incredibly similar to these graphs, but they're showing something completely different. So I'll explain that in just a minute. This is actually a very messy and confusing graph, so I'll just explain what it is. Don't make graphs like this. It's a really nice paper, but I don't like this graph. So they said, okay, we're going to take our fMRI measures, and remember DTI stands for diffusion tensor imaging. It's the measure of connectivity, white matter connectivity. So he said, which of these voxels, which parts of the brain actually end up contributing to the ability of our classifier to figure out does somebody belong to the gets better over 2.5 years group or the doesn't get better over 2.5 years group. And you may remember from last week that we talked about, sorry, on Tuesday, we talked about how your standard reading areas, your standard language areas in general, these sort of left hemisphere, Broca's, Wernicke's type areas. But dyslexics often show a sort of compensation by maybe bringing in right hemisphere areas that don't typically get used otherwise. So they actually found that the areas that were most predictive were these right hemisphere, sort of a more or less mirror image of Broca's area sort of regions. So it's almost as if the more you bring that region into play, the more likely you are to compensate and get better. And so what this is a plot of is activation in this area or white matter integrity in this area plotted against how much people get better in their reading in this case, or in this case it's the two brain imaging measures plotted against each other. But this is kind of a horrible graph because it has two completely different scatter plots sitting on top of each other. So pretend for a minute that you're only looking, pretend that the blue stuff isn't there. Okay? So if you look at the blue stuff, if you look at the red stuff, then you'll see a bunch of dots sort of going up a line with a positive slope. Okay? Just don't draw two scatter plots on top of each other. It just gets messy. Okay? But so mentally subtract away the blue stuff, and you'll see, there's red things going upwards. So in this case, it means the greater your reading gain. I don't know if you can read. You probably can't read these axes labels, but this says reading gain, and this says right inferior frontal gyrus fMRI activation. So if you're a red person, red is the people who got better, the more you gained in reading, the more activation you had here. And this shows the more white matter integrity you have there, the more activation you have there, so the functional and structural sort of go together. And this one shows the more reading gain you have, the more white matter integrity. So this shows that the behavior and the structural connectivity measure will go together.
Raj,
So, yes.
Sorry, quick question.
Yeah,
so
yeah.
the white matter integrity is just, it says nothing about sort of the connections between the right IFG and wherever it is connected to, but it's just saying the diffusion index.
That's actually a good question. You'd have to look at the paper to see exactly. Yeah. I think they measured this. This stands. This stands for superior longitudinal fasciculus, I think, SLF. So I think it was measured along some tract, like this. What is sort of the tract that goes from front to back. But you'd have to look at the methods to see exactly which regions they. It's probably in the supplementary information exactly what they were sort of averaging over there. But it's some white matter tract that sort of connects this region in a front to back sort of way. Okay, so the red stuff goes up, and now pretend that the red stuff is not plotted there, and mentally subtract that and just have this blue stuff, and the blue stuff doesn't go up. Okay, you've got all these sort of flattish, it's really hard to see, but you've got all these sort of, the red things are sort of stretched out clouds, and the blue things are more sort of round clouds. So there's not really much correlation between these measures in the people who didn't improve, but there's a positive correlation in the people who did improve. So that means that this right frontal thing that the classifier found is actually doing what it kind of ought to do. So then what they did was they said, well, how about if we try and quantify how well we can predict using different measures. So what if we use fMRI from this frontal region, diffusion tensor from this sort of right frontal region, combination of both of them, Bx stands for behavior for some reason, and fMRI pattern means if you use the whole brain. And these people are based in the medical school, and they're sort of partly writing for doctors, and doctors like to use these measures called sensitivity, specificity, and positive predictive value. They are actually useful measures, but they get used a lot in medicine. So if any of you are interested in going to medical school, you'll learn these things till you're sick of them, but they are actually useful. So I just want to quickly recap what they are. We did talk about this a little bit back in the day. So this is just pulled off from Wikipedia. This is actually one of the, it's actually quite a good Wikipedia page, I think. And it shows what sensitivity, specificity, and positive predictive value are, except here it calls it precision. It turns out precision and positive predictive value are the same thing. So this is a little bit confusing, but let me just kind of talk you through a bit. So what intuitively would sensitivity mean? Intuitively, sensitivity would mean if something really is there, can you catch it? Right? And so these are really, although these are slightly confusing terms, they're good terms because they actually sort of mean what they ought to mean. Okay, they're not terms like type 1 error and type 2 error, which are horrible terms because you can never remember what on earth they mean. Their meaning is sort of carried in their name. So sensitivity means if something really is there, can you catch it? Right? So it means of all the things that are really condition positive, in other words, of all the, say all the patients who actually do have the disease, how many of them do you pick up with your test? Or in this particular case, it's of all the people who actually did improve in their reading, how many of them did we pick up? Okay, so sensitivity sort of makes sense. Specificity, again, is also sort of, the name carries its intuitive meaning to some degree, maybe a little bit less than sensitivity. So it says, of all the people who don't have the disease, how many of them do we correctly say, yeah, they don't have the disease? Okay? So of all the people who we ought to exclude, how many of them do we successfully exclude? So you can see why that's specificity. So, for a disease, well, I just said what it was for a disease. In this case, it's for all the people who don't get better, whose reading doesn't improve. How many of them do you say, yeah, their reading doesn't improve? And then the positive predictive value, or precision it's labeled here, says, for all the people who your test says, yeah, I think this person has the disease, how many of them actually really do have the disease? Okay? Or in this case, for all the people of whom you say, yeah, my classifier says I think that their reading should get better, how many of them does their reading actually get better? Okay, so these are all different ways of thinking about kind of different aspects of the same thing. Those of you who do psychophysics will use a measure that sort of combines two of these together called D prime, which in my opinion is actually a better way of doing it, which basically says sort of if you've got two distributions, how far apart are those distributions in terms of the difference between the means divided by the variance, divided by standard deviation, actually. So which is kind of maybe a better way of it has various advantages. One reason why people care about this kind of thing is because it's really easy to make a test incredibly sensitive. It could just say, yeah, everybody has the disease. You now have the world's most sensitive test, but it has very poor specificity. Okay? And similarly, you can make an incredibly specific test. It says, yeah, nobody has the disease, but it has very... So at the D prime measure, I'm not going to talk about that here, but the D prime measure sort of like gets around that problem a bit. But anyway, so you can see why this would be of interest from a sort of medical point of view, because it comes up a lot in actual sort of diagnosis. Anyway, that was all just to kind of help explain a little bit what's going on in this figure. Why does it have 4 different dots instead of just showing accuracy, which is what you would often see? So the real punchline here is, okay, that fMRI does, predicting just from fMRI, just in this sort of right frontal region, does reasonably well. Okay, it gets about 60% correct. And if you're interested, you can look at the specificity and sensitivity and blah, blah, blah. Diffusion tensor imaging does almost as well, but not actually that well, and it turns out to have much lower specificity, which I guess means that it quite often says maybe somebody gets better, but it's actually wrong, I think. Is that what it would mean? I think so. If you combine them together, you do a little bit better than if you use either of them separately, which is what you would hope for. That sort of shows that even though they're correlated, they're not telling you exactly the same information. They're telling you related but slightly different information. If they were telling you exactly the same thing, combining them wouldn't help you at all. This is probably the most interesting bit. Behavior. If you just use behavioral measures, you don't do as well. You get about 60% correct, but you don't do quite as well as if you combine both your neuroimaging measures. Okay? So you get something beyond what you get from behavior. That's where the imaging earns its keep. Although it's worth pointing out that actually if you just use behavior, if you compare this 4th column to the 1st column, as if you just use fMRI just from this one region, you do about the same. There was a very interesting case recently of people are quite into releasing these sort of public data sets, and there was a big public data set released of ADHD resting state data, and there was a competition to who can predict the best from this, who has ADHD and who doesn't. And it was really beautiful because the people who won actually didn't use the imaging data. They just used the behavioral data. I think they may have subsequently done some studies showing that if you combine both of them, you do even better. But it was just it was very cute. I'll try and pull up the reference for that. But it was very nice and very interesting, and it shows that it's really worth asking that question. Okay. Now I think I hesitate. I may be remembering this wrong, but I think they actually did do a follow-up that showed that if you combine the behavior and the brain data in the right way, then you actually do a little bit better. So it's not saying the brain data is, this is not saying brain data is useless. What it's actually more, it's more saying kind of the converse, behavioral data is really useful. Okay, but the brain data and the behavioral data are probably not telling you exactly the same thing. So if you use both of them, you're probably going to do even better. Okay, now those of you who know about SVMs are probably wondering, well, why did they have to split the data up into this, in this kind of somewhat artificial way of saying, these are the people who got better, these are the people who didn't get better, can I tell them to apart? Why didn't they use regression, or in this case SVM regression, to actually say, the degree to which people got better is actually a continuous variable, why don't you try and fit that variable, using the brain data, using this thing SVM regression, or just regular regression, in fact. And the answer is maybe they should have done that. It probably would have been a reasonable thing to do. But this graph actually shows that if they had done that, it would have worked out okay. So just to go back to here. So you've got these two classes. This is true for any classifier. This is not just true for SVMs. So you've got these two classes, and the classifier says, okay, give me a new point. I'm going to tell you whether I think it's blue or red. Okay? Well, as you might imagine, points which are really, really, really far away from the boundary, in some sense the classifier is much more confident that they're blue or that they're red. Whereas ones that are sort of close by, it's like, yeah, I think it's blue, but I'm not that confident about that. Okay? So it's a very simple measure of how far is something from the boundary. Is sort of a measure of like how well are you classifying it. I mean, in a sense, how well are you classifying it is just a kind of binary, yeah, is it right or are you wrong sort of thing. But in fact, the further away it is on the correct side, the more right you are, right? And the further away it is on the wrong side, the more wrong you are. And in fact, actually, that's sort of how, for those of you who are wondering, what do you do when the clouds are overlapping and there is no point in the middle that has an actual clean margin about it? What you do is you actually build in a measure, they call them slack variables, where you sort of encode, how far away from the boundary of things are if they're on the wrong side. That's a whole separate topic, but anyway, that's what, if you ever heard the term slack variables in SVMs, that's what it means. So all you need to take away from that is that the further away something is from the boundary on the correct side, so if you say that you think something's blue and in fact it's way over here in the blues, then you're doing really well. That's a sort of measure of how successfully is the classification happening, even though it's really a sort of right or wrong, thumbs up, thumbs down sort of thing. So that's expressed here in this term distance from hyperplane. Hyperplane is just the sort of jargon term for a boundary that's in more than 2 dimensions, or more than 3 dimensions, I guess. So the further something is away from the hyperplane, the more confidently it's classified by the SVM. And that's true not just for SVMs, that's true for any classifier. So this is a plot of behavior. So the more someone gained in reading, the more confidently the SVM classified them. So that's good. So that means that it's sort of getting the right answer, and it's sort of getting the right answer in a sort of sensible way. And it also means that if they had actually done SVM regression, they probably would have gotten very similar results. So I like this study a lot, as you can probably tell, and for multiple reasons. First of all, because dyslexia is just, for me at least, a kind of interesting topic that certainly matters to a lot of people who have it, and families of people who have it. But it's also kind of very interesting because it uses classifiers and it does something that shows that you go beyond behavior. So the imaging sort of earns its keep. Now, what's the most extreme case that you could possibly imagine of going beyond behavior? The most extreme case is when there is no behavior. You can't get more extreme than that. And it turns out that people have actually done some very interesting studies of looking at brain activation in people who do not have behavior, okay, but who might have something going on in the inside. So I often talk about, what if people look the same from the outside but might be different from the inside? Well, what's the extreme example of that? And this is sort of terrifying, it does happen to people. If you have some terrible brain injury and you become completely paralyzed and you can't move and you can't express yourself and you can't respond to anything, but there's something going on inside. You're locked into your body. Terrifying, terrifying prospect, but it happens to people. Okay, and I hope you're not too queasy because I'm going to show you some pictures of really kind of stoved-in brains. Just MRI scans, there won't be blood or anything. To me, I actually feel a bit queasy when I look at those pictures. So, trigger warning, you're about to see some pictures of sort of, highly damaged brains. But it's just, it's just a 2D slice of MRI. It's not that bad, really. But one reason, so there's multiple reasons why this is interesting. And I'm about to show you a BBC News video about this. It's gotten a lot of attention, rightly. So one reason is because obviously it's clinically important, right? But another reason why I kind of like it, well, this is a sort of reason why you might not like it, but in my opinion, you should like it, okay? So what they do is unbelievably, almost boneheadedly simple, and I mean that as a compliment, and they would be the same. It's really, really, really simple. It's taking an incredibly simple idea that you might think, oh, this is not sophisticated at all, but it's really important. Okay? And in fact, if they were trying to do it in a really sophisticated way, it would be much less believable. Okay, so I'll show you in a minute what they do, but anyway, let's watch this video. It's just a short video. I mean, there have been entire hour-long documentaries made by the BBC about this. The guy who did it, called Diffusion, and his colleagues, was at Cambridge, and now he's at University of Western Ontario, which is not very far from here, actually, near Toronto. Okay, so let's watch this video. So hang on a minute. Okay, vegetative patient Scott Routley says, I'm not in pain. This is just from BBC News website from a couple of years ago. No, a year and a half ago.
Okay. Right, this is your big chance. We really want... Scott's on his way to the University of Western Ontario. He'll meet Professor Owen, who moved to Canada last year, but still collaborates with colleagues in Cambridge.
Big loss for UK science, actually, won't you like? Big win for Canada. Oh, really?
Scott, right, this is your big chance. We really want you to try as hard as you can to imagine playing tennis for us. Okay, we're going to ask you to imagine playing tennis quite a few times while you're in the scanner. And if you can do that, then we'll be able to see your brain activating. So I want you to imagine you're standing on a tennis court and moving your arm around as much as you can. All right, so we're going to put you in the scanner now. Just do your best. Stay awake.
Can you hear this okay?
Can you pause this? Can I ask a question?
Yes.
So blinking is behavior? Or?
Yes, that's a very interesting question. So there's different degrees of kind of locked-inness. And so for instance, if you're very unfortunate and your spinal cord gets broken in your neck, which unfortunately happens to quite a few people, then you can't move anything from below the neck, but you can sometimes move your face, maybe in very limited ways. So sometimes these people, for instance, can only blink. And there's actually a famous book, I haven't read this book, because it hasn't come out as an audiobook, but called The Diving Bell and the Butterfly. You ever heard of that? It got made into a movie, actually, which apparently was quite good. Which is an entire, I think the guy was like an editor at French L or something, so he was a very accomplished writer, and then he had this, I think it was a diving accident or something, and he could only blink. Okay? And he wrote the entire book just by blinking, so some very, very patient caregiver, I don't know what the code was, maybe he blinked in Morse code or something, I don't know. But all he could do was blink, and he wrote. So there's that's kind of sort of locked in, but these people are even more locked in than that. Okay, they're so locked in that it's an actually genuinely open question. Well, in fact, it wasn't even an open question. For the longest time, they were just categorized as, they're given the label vegetative state. Okay, that's not a very nice label, right? I mean, you give the label vegetative state when you basically say, there's nothing going on here. So it's different. I actually didn't realize this until recently. It's different from being in a coma. Being in a coma is, like when you're just asleep, okay? But being in a vegetative state, so you saw actually the person, his eyes were open and he was sort of wiggling a bit and stuff. So he's not asleep, but you can, shout at him, tickle him, do whatever you want, and he's not going to respond to you, or he'll just respond. This is actually what makes it particularly hard to diagnose. I mean, a doctor could, I'm not a doctor, but it's very difficult clinically because a tickle may actually be, I may have said something wrong there. Basic reflexes are still present. Okay, so in fact, if you tickle someone, they might kind of flail their arm or something. Okay, or if you, poke them, they might sort of withdraw their hand or something, and you might think, oh, they must be conscious. But you can't get them to do anything voluntary. If you say, please wave your hand if you want to say hello, or this very hand they just moved when you tickled them, they can't do it, or it might look like they do it, but you can't do it reliably. So to all intents and purposes, there's nothing you can get out of these people. So you might say, okay, there was actually, from a behaviorist, 1920s psychology point of view, there's nothing more to it. That literally means there is, there's no mental processing happening. But you might think, well, hang on a minute, maybe there is something going on in there, but we just can't get to it. Well, this is trying to get to it. So now I've forgotten what your question is.
About the blinking. I
Yeah.
just didn't know vegetative state.
Yeah, so no, these people cannot communicate by blinking. Yeah. And there are people who communicate by blinking, but they're classified differently. So they would be called, I don't even know what the correct clinical term for that is, actually. Does anyone know that? Yeah.
It's more than quadriplegic.
Yeah, it is. I don't know what the right, quadriplegia just means you can't move any of your 4 limbs. Yeah, I don't know. That's a very interesting question, because often these people are referred to as locked-in patients. But if you can blink, then you're kind of sort of not locked in, but you can't do much. Yeah, I don't know what the right name is for that. So anyway, but these are people who are even more locked in than that. So locked in that it's actually an open question of whether there's anything inside. And in fact, well, I'll tell you the punchline now. They found reliable sort of communicative brain activation in only about 20% of them. Okay, so for most of the people who are described as, there's nothing going on, it's actually correct. But for a very non-trivial number of them, they actually have found, hey, hang on a minute, there is something going on. Yes?
Well, might it be possible that we're not able to detect the responses with this particular method?
You mean like maybe if they'd done the right behavioral test, they might have found it?
Yeah, or a different, more sensitive scanning technique. Or
well, very good question. We'll talk about that more.
Okay.
Yeah, but that's entirely possible. It's certainly in terms of the non-scanning stuff that was done, like in the paper, this Monti et al. Paper, it actually talks about, some of these patients were evaluated by a highly trained team continuously for an entire month. And they still couldn't figure out whether there was anything going on there. I mean, it's a miracle of A, medicine, and B, how well people who are, in a proper healthcare system get treated. That, there's someone who's just, in the bed can't do anything, and their family still cares for them. Teams of doctors still try to look after them. I mean, this would not have been true for most of human history. Okay, so, let me just go back in time. Okay, so they're about to put the guy in a scanner. And just imagine how weird it must feel talking to one of these people. Okay, I mean, this poor guy, right? He's just lying there. And at this point, you have no idea, is he one of the 20%? They didn't know before they did the study that nobody had ever shown that any of these people had anything going on inside, right? So this is kind of why this is a big deal. But, their families still, have birthday parties for them and stuff. I mean, it's really amazing, the commitment that people have to their loved ones. And so imagine what it must feel like talking to someone when you just literally have no idea at this point, is there anything going on inside?
Scott's on his way to
sorry.
the University of Western Ontario. Playing tennis quite a few times while you're in the scanner. And if you can do that, then you'll be able to see your brain activating. So I want you to imagine you're standing on a tennis court and moving your arm around as much as you can. Alright, so we're going to put you in the scanner now. Just do your best. Stay awake. Hello, Scott. We're going to try again. We're going to try and get you to imagine playing tennis.
Scott is such a clear responder, they decide to ask a question that no patient in this position has ever been asked before. A question which could have profound consequences.
I'm going to ask you a question about you this time. A question about you and the way that you're feeling. And I want you to try and answer this question for us. I want you to tell us whether you are in any pain. Think about your body. Does any part of your body actually hurt right now? Are you in pain?
Right, so what does that tell us that he's imagining trying to answer?
It tells us that he's imagining playing tennis. I've told him to imagine playing tennis if the answer to the question is no.
So that would suggest he's not in pain.
It would suggest that he's not in pain, which is a big relief.
Have you ever asked that question before of any patient?
No. No.
That must have been quite a nerve-wracking moment, right? Matt, what if he said yes? Right? 'Cause, I mean, this raises a lot of interesting questions, which we can talk about in a minute. Okay, but I just want to.
I have a question.
Yes.
I missed it. How did they know that his answer was no?
I'm going to show you that in a minute. Yeah, it that didn't explain that sufficiently, so you were right to miss that. Okay, so a couple of things it's important to know here, which were not sufficiently explained in that 2-minute video. Okay, so first of all, a little bit about imagery. So there used to be, I think this is actually one of the very few things in cognitive neuroscience where there was sort of a debate and it sort of got answered. Okay, usually that doesn't happen. Okay, but in this particular case, I think it's not ridiculous to say there was this whole debate and then they figured out the answer. And so this guy called Steve Coslin, who was sort of on the right side, the winning side of that debate, that he gets to write the Nature Reviews Neuroscience article about it. So one school of thought said, imagery, well, the more intuitive one is the imagery is basically like a sort of weaker version of the actual process. So if you're perceiving something visual that lights up your visual cortex, if you imagine seeing something visual, then according to this hypothesis, you'll sort of light up your same area, but a bit less. Now, of course, before brain imaging, it was very, very difficult to answer that question. Same for motor. Now, you just saw a motor test. They was asking people to imagine playing tennis. So here's an example from this Nature Reviews Neuroscience article. You imagine that you're actually turning in this case,